All right, so we're going to be starting chapter 3. This is the molecules of cells. So as an introduction, if you consider the sugar lactose, which we find in dairy products, you'll be surprised that the bulk of the world's population isn't able to digest milk-based foods because their body is not able to break down lactose. We're talking about roughly 80% of African Americans and Native Americans. That number climbs to about 90% amongst Asians. The reason is their body either does not produce any, or it produces very little of the enzyme that actually breaks down lactose, which is called lactase. So if your body is not able to break down lactose, then you have this condition called lactose intolerance. So again, this shows how important biological molecules are in daily functions of any given living organism. So when you look at organic compounds are carbon-based molecules. They contain, they all have carbon in common. That's the main thing. They'll have carbon. You'll find nitrogen in them also, hydrogen. Sometimes they'll have nitrogen, sometimes they'll have oxygen. But what they always have is carbon and hydrogen. You'll see that in them all the time. Carbs are primarily talking about sugars. So when you look at honey is a type of sugar that bees make. It's one of the healthier sugars. Fats or lipids. Fats are this type of lipids, and lipids play all kinds of roles. Not only do they make fats, but they also produce hormones. We'll come back to that. Proteins also. The building blocks of proteins also produce enzymes like lactase, for example. So again, lots and lots of different functions for both lipids, proteins, carbs, these organic compounds, and also nucleic acids. We're going to be talking about these briefly. This is essentially, when you look at it, this is a double strand. It's a DNA molecule, and essentially because of nucleic acid, proteins are produced. When you look at this molecule over here, this is that of lactase, and what do you see? Kind of to a normal person, since most of you have not studied or probably haven't studied any biochemistry at this point, this kind of probably looks a little bit disorganized, but it's actually not. It is organized. These ribbons. Because of the way that this is structured, this is able to do what it needs to do, which is break down lactose. So as far as organic compounds go, so life's molecular diversity is based on the properties of carbon. Diverse molecules found in cells are made up of carbons that are bonded to other carbons and or atoms of other elements. So carbon-based molecules, we refer to them as organic compounds. Now, this happens by the carbon sharing the electrons. Now, the electrons, it can bond to 4 other atoms, and it can branch into up to roughly 4 different directions. Methane, for example, CH4, this is one of the simplest organic compounds. It's just 1 carbon atom, and it has 4 bonds, single bonds to hydrogen. Now, the type of bonds that you find in methane are covalent bonds. Now, remember, these are sharing. So carbon, it has on its outer valence, in its outer shell, it has 4 valence electrons. So that means, remember, the second shell can fill up 8 electrons. So it needs 4 more to be completely happy. So that's why it's very happy to take on 4 hydrogen atoms. So each of the 4 lines that you see in the formula for methane, it represents a pair of shared electrons. Methane and other compounds composed of only carbon and hydrogens are referred to as hydrocarbons. So a lot of the fuels that we have, they're hydrocarbons. Propane, for example, is one of them. Methane, again. Butane, it's another one that is what you find in your lighter fluid. So carbon with attached hydrogens can bond together in chains of various lengths. So again, if you look over here at this, these drawings here, so this over here is what we refer to as a structural formula, and essentially this shows you a single carbon atom, and each one of these lines represents a bond, a single covalent bond. So you have one covalent bond to a hydrogen, another one here, another here, another here. So you have 4 hydrogen atoms and 1 carbon atom. Now this is a ball and stick model, and what this allows, again, it's pretty much showing you the same thing. That you have that 1 carbon atom, and then you have the 1, 2, 3, the 4 hydrogen atoms, and these little sticks, they're the bonds. And this shows you the type of bond, which is a single bond. And also, this red line, it's showing you that these hydrogens, they're bonded in a, almost like a type of a pyramid, triangular pyramid type of arrangement. And this is a space filling model. Again, you have the black over here is the carbon atom, and then you have the 4 hydrogen atoms. So carbon skeleton is a chain of carbon atoms that can be branched or unbranched. So compounds with the same formula but different structural arrangements are called isomers. And we'll look at examples of this next. So when you look at the carbon skeleton, this is what we're talking about. Carbon, 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 just these little carbon, this area that's shaded in the gray. This is your carbon skeleton. What comes off of these, we're going to be talking about in the slides to come. So when you look over here, this is what we call unbranched. In other words, they're linear, the carbon atoms. You got one here, another one here for ethane. Then when you have propane, you have 1, 2, 3 carbon atoms that are linear. Now, when you look at branching, this is an example of a branching structure. So you have 1, 2, 3 carbon atoms that are going straight, but off the second carbon atom, you have one that goes up. So 1, 2, even though this has 4, and this also has 4 carbon atoms, notice the formula is the same, the molecular formula is the same, but the way that it's arranged is what's different. So this is what we refer to as these isomers. Something else that can happen, again, that'll affect the shape and the angles, and also in return, it changes the entire function of this compound, will be bonds. So, and where these bonds are found. So over here, between the first and second carbon, you have 2 lines. This is a double bond. Now, so we call this 1-butane because it happens at the first carbon. This is at the second carbon, 1, 2. So we call this 2-butane. Again, when you look at it structurally, it's going to look very different. If you look at it at the ball and stick model, this would look very different than this, which would look very different than that, and this would look very different from this. Even though their the chemical formulas are the same. They all have the same number of carbons, and they all have the same number of hydrogens. Again, just the way that they're arranged, it affects what they look like structurally and their function in living organisms. Rings. Sometimes you'll have these organic compounds formed in rings. So, for example, this is called cyclohexane. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Sorry, 1, 2, 3, 4, 5, 6. You have 6 carbon atoms over here. That's hex. Hex means 6. If there was 5, we would call it pentane. Again, when you look over here, the difference are the bonds. Notice the double bonds over here, there, and there. So cyclohexane, benzene. And it's all, again, the formula is the same, but what do we have different? I'm sorry, the formula is not the same over here. What do we have different though? We have these double bonds that are different. So a few chemical groups that are key to the functioning of biological molecules. So an organic compound has unique properties that depend upon the size and shape of the molecules and groups of atoms, or functional groups that are attached to it. So a functional group affects a biological molecule's function in a characteristic way. In other words, how it interacts with other molecules. This is what this functional group is about. We're going to be looking at 5 functional groups. Actually, yeah, we're going to look at roughly 5 functional groups, and then we're going to look at one more. A total of 6 groups in the slides to come. So compounds that contain functional groups are what we call hydrophilic, or they're water-loving. Now, in the organic molecules, organic compounds, organic molecules, we also have functional groups. Let's look at these first 5. 1, 2, 3, 4, 5 over here. Now, these 5, they're hydrophilic. functional groups. So hydroxyl groups, they consist of hydrogen that's bonded to an oxygen molecule, or that are bonded to an oxygen molecule. Carbonyl groups will have a carbon that's linked by a double bond to an oxygen atom. Carboxyl groups, they're made up of carbons that are double bonded to both an oxygen and a hydroxyl group. Now in an amino group, you'll have carbon atoms, or you have a carbon skeleton that has nitrogen that's bonded by 2 hydrogen atoms. In the phosphate group, phosphate groups now are made up of phosphorus atom bonded to 4 oxygen atoms. Okay. So let's look at the first one, the hydroxyl group. And over here in alcohol, you see you have these 2 carbons, and then you have this functional group right here, this hydroxyl group. In the carbonyl group, again, what do we have? The carbon and double bond to an oxygen. So in the aldehyde, you'll see that the carbon is all the way at the end. This is the number 3 carbon, where you have this carbon double bonded to the oxygen. In the ketone, you have in the number 2 carbon, the carbon that's bonded to the oxygen, double bonded to the oxygen. Oh, excuse me. So again, this is the functional group. So when you have a chemical reaction, the exchange will take place over here between this carbon, this oxygen molecule, or in this case, in the hydroxyl group, between this oxygen and hydrogen over here. In a carboxyl group, again, you have a carbon, 2 oxygen, and a hydrogen molecule. So if you look over here, that the carboxylic acid, it's the number 2 carbon over here that has the double bonded oxygen over here, and then you have this hydroxyl group over here. And again, when it gets ionized, what's going to happen is the hydrogen gets lost, and again, you have this slightly negative. This molecule becomes slightly negative. In an amino group, you have nitrogen, and then you have the 2 hydrogens. So when you look at the amine, again, you have this nitrogen and 2 hydrogens. Now, when the amine becomes ionized, you end up gaining a hydrogen. So again, it's slightly more positive now. The phosphate group over here. So again, what do we have? When you look at here, this is the phosphate group. Again, you have this oxygen, and then you have the phosphorus and oxygen and 3 oxygen molecules. So when you look at adenosine, when you look at this, so this is what we'll refer to as the organic phosphate: oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus, and then again you have your oxygen molecules that are branching off of it. So here we go. 1, 2, 3. Your phosphorus over here, and then your oxygen over here. Now the methyl group over here. This is not a hydrophilic functional group. So this CH3 over here in this methylated compound that we see right over here, CH3, this is what makes this compound methylated, because this methyl group over here. Okay, the chemical reaction will take place over on this end. So an example of similar compounds that differ in functional group is a sex hormone. So male and female sex hormones differ only in the functional groups. Everything else is the same. The difference is they cause varied molecular reactions. So the results, the distinguishable features of males and the females. So when you look at the lion, the male lion has this hormone, testosterone. And if you look at the female hormone, estradiol. Now look at where the difference are. Again, it's in these functional groups. In estradiol, in this case, you have this hydroxyl group. In testosterone, you have a carbonyl group instead of the hydroxyl group. And in addition, you have this methyl group over here. So this causes the effects, the physiological effects are apparent in this male lion, and the physiological effects from estradiol are apparent in the female line. So cells make a huge number of large molecules from a limited set of small molecules. And essentially we have 4 classes of molecules that are important to organisms: carbohydrates, proteins, lipids, and nucleic acids. So the 4 classes of these biological molecules, they contain very large molecules. So they are often called macromolecules because of their large size. They also could be referred to as polymers. The reason we call them polymers is because they're built from smaller compounds, smaller molecules called monomers. So again, the building blocks of these polymers are the monomers. So monomers, they link together. In other words, they come together to get bigger and bigger and bigger to form polymers by chemical reactions that's referred to as dehydration reactions. Okay, and essentially what happens is the water gets removed from it. So if you think about when you're dehydrated, what happens? You don't have enough water in you, right? You sweat through. For example, on a very hot day, you're outside, you're doing work, you sweat a lot. In other words, you're getting rid of a lot of water, so you're dehydrating. So the same thing in chemical reactions. When you lose water, dehydration occurs. Monomers, they come together to build polymers. Now, if you want the opposite to happen, in other words, you want to take these polymers and break them down, make them smaller, you have to add water. And this process is called hydrolysis. So when you look at this word, hydro, again, you have water, lysins to break apart, hydrolysis. You're adding water to break up this larger molecule into smaller monomers. An easy way to remember this, think about some of you, if you have a basement and if it gets flooded, what happens as all the water starts to rise, it gets into the wood, it's going to break the wood apart. So if you have wooden cabinets, the water as the flood water, it gets into the wood and then it starts to break it apart. So all biological reactions of this sort, they're mediated by enzymes. Enzymes, again, these are proteins that just speed up chemical reactions. They help the reaction go through faster. So a cell makes a large number of polymers from a small group of monomers. So, for example, proteins. There are only 20 different amino acids, and the quantity and the way that these amino acids are arranged make all different types of proteins that we find in our body, for example. So DNA, on the other hand, comes from only there's only 4 base pairs, 4 kinds of nucleotides, okay? That's responsible for making DNA. Monomers are used to make polymers are universal. Yeah. Now, let's look at an example. So over here, you have this monomer over here. Okay, it's unlinked, it's all by itself. And you got the short polymer over here. Okay. Now, what's going to happen? This hydroxyl group over here, okay, it's going to react with this guy over here, this hydrogen here. In other words, this hydroxyl group is going to go away through dehydration, and it's going to link up with this hydrogen here to form water, and then this chain over here is going to link with this chain here. So if you look over here, essentially what happens is, again, this guy here, they came together and left this guy all alone. They left it open. So what ended up happening is, this guy over here, now that it didn't have a hydrogen, it wanted to bond. So it went and it connected with this this chain over here. And you end up having this longer polymer down over here. Now, what do we do if you want the opposite to happen? Okay, we got this long guy over here, 1, 2, 3, 4. We want to break it up. Remember, what do? Hydrolysis. We add water. When you add water, what's going to happen? We add water. You have OH over here. Okay. So this guy over here, when you add the water comes in, it breaks it over here at this bond over here, and the hydrogen, one of the hydrogens will go attach itself over here, which is what we see over here, and then what we have left now is 1 oxygen, 1 hydrogen, a hydroxyl group, and that will go and attach itself, this part over here to this end. So carbohydrates range from small sugar molecules, which are called, again, these are the monomers, to large polysaccharides. So starch, for example, that's an example of a large polysaccharide. Sugar monomers, we refer to them as monosaccharides, okay, or single sugars, small sugars. You find these in honey. So, for example, glucose and fructose. Okay, this is an example. Monosaccharides, they can be hooked together to form much, much more complex sugars, and also polysaccharides. So again, this is an example of the honeybees. They're producing honey. Again, it's a sugar. It's one of the healthiest sugars you can eat. The carbon of skeleton of monosaccharides vary in length. So glucose and fructose, they have 6 carbons. They're only 6 carbon longs. Others, they could have 3 to 7 carbon atoms. Monosaccharides, they serve as the main fuels for all the cellular work. In other words, they help all this is the gas that fuels the cells in our body. Okay, these simple sugar monosaccharides. They're also used as raw materials to manufacture other organic molecules. So if you look over here, glucose, and you have fructose over here. And what do we have? This carbonyl group on the fructose, that's at the number 2 carbon. And for glucose, you have the carbonyl group at the number 1 carbon. So many monosaccharides are also form rings. So the ring diagram may be abbreviated by not showing the carbon atoms at the corner of the rings, and they could be drawn with different thicknesses for the bonds. This will indicate the ring is a flat. It's a flat structure that's attached. Atoms extend above and below it. So if you look over here, let's look at the structural formula here. It shows the carbon, the number 1, number 2, 3, 4, 5, 6 carbons here. And when you look at the abbreviated structures, again, the numbers are gone, everything else is there. All right? So, and then you look over here, the simplified structure. You don't have all the O's and H's that are displayed, but they tell you where the oxygen is. That's it. All right? So when you start studying organic chemistry, you guys will be familiar with looking at these structures. Something like this, and you'll know exactly what, just by looking at this, you'll know that what it means. So 2 monosaccharides are linked to form a disaccharide. So when 2 monosaccharides, or 2 monomers, can bond, they form a disaccharide, okay, in the dehydration reaction. Again, when you look at this, di means 2. Okay, mono means 1. So mono, di, 2 saccharides, 2 sugars. So disaccharide sucrose is formed by combining 1 glucose monomer and a fructose monomer. Okay, so sucrose, this is like your table sugar, for example. Again, it's just glucose and fructose that comes together. So disaccharide maltose is formed from 2 glucose monomers. So again, what do we have? You have a glucose monomer and a glucose monomer over here. And now we want to form a polymer, a bigger chain, right? So what's going to happen? Dehydration or hydrolysis? Can you guess? You want to bring them together. So we want to dehydrate. In other words, we want to get rid of the water. So this OH here and this H, they'll come together to form water, and then this oxygen is going to link these 2 sugars together. There we go. Water is, take away the water, and then there goes that oxygen, and you end up getting maltose. So polysaccharides, again, these are big molecules, okay, macromolecules and polymers that are made up of thousands. They could be made up of thousands of monosaccharides. Polysaccharides may function as storage molecules or structural compounds. Starch, again, I told you, this is an example of a polysaccharide. Potatoes, for example, lots of starch they contain. So made up of glucose monomers, and they're used by plants for energy storage. Okay, so this is what animals do. Animals, they'll take the extra glucose and they store it as starch. Glycogen is what you find in animals. So in humans, for example, you find glycogen as an energy store, a temporary energy store. So glycogen, again, this is also a polysaccharide. It's made up of glucose monomers just as well. Again, however, we use it. So in glycogen in the human body, for example, it gets stored in muscles and our liver. So for example, when if we try to run, just say you're walking down and you're trying to catch the bus and you start running really fast. Well, what's going to give you your muscles that burst of energy to go is the glycogen that's stored in your muscles. Long-term storage, it gets converted into fat. We're going to be talking about that later. So long-term storage, fat. Short-term storage, sugar storage, glycogen for humans. Cellulose, okay, this is also a polymer of glucose, and you find this in the cell walls of plants. Chitin, this is also polysaccharide. Now you find this in insects and in crustaceans, and it helps build their exoskeleton. Our body uses also in healing process as well. So again, when you look over here, what do we have? You have these glucose monomers. So starch, again, just have this long chain, single chain polymer over here, starch. Okay. Now, and they're showing you the example of potatoes. Potatoes have lots of starch, which is why if you want to lose weight, you don't want to eat lots of potatoes. Glycogen, on the other hand, again, you have the same thing. You got this glucose monomers. They're linked together. And however, what ends up happening is they have branches coming off of it. Again, so a little bit different than what the starch looks like. Then you have cellulose, again, which you find in the cell walls of the plants. Again, you got these glucose molecules. However, what do you have? All these molecules, they get bonded between them. So you got hydrogen bonds between these glucose monomers in rows, in columns, I guess. So polysaccharides are usually hydrophilic. In other words, when you see hydro, again, we're talking about water. Philic means loving, so water loving. Bat towels, they're made up of cotton. Cotton, it's mostly cellulose, and what do? They like water, so they're water absorbent. Now we're going to be talking about fats, we're going to be talking about lipids. Examples of lipids. Lipids do make fats. So lipids, they're water insoluble. Okay, in other words, they're hydrophobic. When you see phobic, it means scared. So they're scared of water or water-fearing compounds. They're important in long-term energy storage. So again, if you eat a lot, if you're, for example, if you're having lunch or breakfast for that matter, or dinner, doesn't make a difference what. But if you overeat, that extra sugar, it ends up getting. First thing it'll end up doing is getting converted into glycogen, which will get stored in some in your liver and then the rest in the muscles of your body. Now, if you keep on eating, even after that, the glycogen is made because your liver and the muscles in your body, it can only hold so much glycogen. Now, there's a limited amount of room that for glycogen to be stored. But if you keep eating, now what's going to happen? Your body can't just continue to make glycogen because there's nowhere to put it. So what ends up happening is your body makes fats. Okay, and that's long-term energy storage. So in other words, it makes lipids up. The extra sugar will get turned into lipids, and specifically triglycerides for our case. So lipids, they contain twice as much energy as polysaccharides, and it can consist mainly of carbon and hydrogen atoms linked by non-polar covalent bonds. Okay, this is one of the key difference: non-polar covalent bonds. When you look at a feather are hydrophobic. The reason they're hydrophobic, or in other words, they repel water, for example, in ducks, is because on the feather there's a very thin coat of oil on there. Okay, and that oil, it repels the water, it allows the bird to stay afloat, and it doesn't allow their feathers to get wet, so they can still fly and float easily. Lipids, they also differ from carbs, proteins, and nucleic acids in that they're not huge molecules and they're not built from monomers. Lipids, they vary a great deal in structure and function. We're going to be looking at 3 types of lipids: fats, phospholipids, and steroids. Okay? Now, a fat is just a really large lipid that's made up of 2 kinds of smaller molecules. And so in other words, what are the building blocks of fats? Okay, they are glycerol and a fatty acid. So a fatty acid can link to glycerol by a dehydration reaction. Fat contains 1 glycerol linked to 3 fatty acids. Fats are often called triglycerides because of their structure. So if you look over here, this is this glycerol over here. Again, it's a 3-carbon in the 3-carbon atom, 3-carbon molecule, glycerol. And then you have the functional group over here, this hydroxyl group over here. And then you have this fatty acid over here. You have the hydroxyl group over here. So what's going to happen? This fatty acid is going to attach itself to this glycerol group up over here. How will that happen? Through dehydration. You're going to get rid of the water. Okay, so the OH from here and the H from there will go form water, and these 2 guys get linked together. and then you got 1, 2, 3. 3 fatty acids come together. So again, we call this a triglyceride. So fats are lipids that are mostly energy storage molecules. Some fatty acids, they contain one or more double bond, forming unsaturated fatty acids that have one fewer hydrogen atom on each carbon of the double bond. This can cause kinks or bends in the carbon chain and prevents them from packing together tightly and solidifying at room temperature. Fats with the maximum number of hydrogens are called saturated fatty acids. Unsaturated fats include corn and olive oil. Okay, these are good stuff. Unsaturated fat. These are the healthy fats. So most animal fats are saturated fats. Hydrogenated vegetable oils are unsaturated fats that have been converted to saturated fats by adding the hydrogen. Not good. Okay, this is unhealthy. This hydrogenation creates trans fats. Again, you get a lot of bad health conditions that result from it. So again, you want the unsaturated fats that you find in olive oil. Olive oil is quite healthy. It's one of the healthier oils, more so than corn oil. Phospholipids, again, when you look at it, what do you have? Lipids and phosphorus. So a structure similar to fats and major component of all cells, phospholipids. They're structurally similar to fats. Fats contain 3 fatty acids attached to the glycerol. Phospholipids contain 2 fatty acids attached to glycerol. So again, fats have 3 fatty... Oh, let's go back here. So fats contain 3 fatty acids, whereas the phospholipid contains only 2 fatty acids, and they're both attached to glycerol. So you look over here, what is this they have? Phospholipid has 2 fatty acids, and the fat would have 3. Now where do you find these phospholipids? In the cell wall. This is what it is, the cell wall over here. So over here, we're going to look at a close-up of this molecule now. So this head and tail, we say the head is hydrophilic. Okay? The head is hydrophilic head. It likes the water. The tails, we say they're hydrophobic. So what ends up happening here is this. This part is okay with the water. This part is okay with the water. But this part here, the tails, they repel the water. Okay? And this is what makes up the cell wall in our bodies, for example. So the glycerol group, again, you get a phosphate group that's attached here. Then you have the 2 hydrophobic, tails, 2 fatty acids. The phospholipid clusters into the bilayer of the phospholipid. The hydrophilic heads are in contact with the water of the environment and the internal part of the cell. The hydrophobic tails band in the center of the bilayer. Bilayer that we find in the cell wall. Right, this is the head right here. So this is the hydrophilic head on both ends. One end over here and the other end over here. And this is the hydrophobic tail of the phospholipid. So essentially what it means is this. The heads, they like the water, so they're going to be facing inside the cell wall and outside the environment that the cell is found in. And channeling between the outside and the inside are the tails, the hydrophobic tails. So these guys are going to be repelling the water away. So whatever is inside, the water that's inside the cells, is not going to be able to escape because these tails are hydrophobic. So they're going to kick it back out, or they're going to prevent it from going out. And same goes for the outside, the environment. The water that's in the outside of the cell, it's not going to be able to go through because of these tails. There is a way that the water comes in, but we'll talk about, you guys will learn about those later. So steroids are lipids, which the carbon skeleton contains 4 fused rings. Cholesterol is a common component in animal cell membranes, and it's the starting material for making steroids, including the hormones, sex hormones that we talked about earlier, testosterone, for example. So when you look over here, this is a perfect example of the sex hormone that we looked at earlier, the estradiol. I'm sorry, no, this is the testosterone. So anabolic steroids, they're synthesized variants of testosterone. They cause a buildup of muscles and bone mass, and they're often prescribed to treat a general anemia and some diseases that destroy body muscles. Anabolic steroids are abused by some athletes, which have serious consequences. If you're not sick, you don't have any health conditions, you should not be using them. Even the people that do use them, that do have some medical disorders, they will all often experience some of the same side effects. They may, which include you get these mood swings, which could be violent, depression, also liver damage is quite common, especially among athletes or individuals that abuse it, because they tend to take so much of it, very high quantities, high doses. Also, they can lead to cancer. Anabolic steroids also lead to high cholesterol and as well as high blood pressure. So again, you need to be very careful. When you are receiving these, you have to be under the supervision of a physician that monitors your body. So again, not a good idea. You want to make muscles like this, you need to do it naturally. Proteins is the next section. So we're going to stop here, and then we'll continue in another series with proteins.